development of seed. So, in dicotyledons, seeds, two cotyledons are there which are laid with lot amount of food materials. That is, cotyledons are fleshy because of storage of excess food materials inside them. Apart from these cotyledons, they contains the embryonal axis. So, this embryonal axis is called as tigellum, and this embryonal axis get attached, or the cotyledons attached to the embryonal axis. The point of attachment of cotyledons to the embryonal axis is called as cotyledonary node, and the portion of embryonal axis above the cotyledons is called as epicotyle, and the portion of embryonal axis just below the cotyledons is called as hypocotyle, and the extreme tip of this epicotyle is called as plumule. So, this is considered to be the first apical part of the, that is shoot apex, first shoot apex, whereas the tip of this hypocotyle is called as radicle, which is considered to be the root apex. So, these are the structures that we can observe inside the dicotyledonous seed. In this, if you see the diagram of this dicotyledonous seed, the two cotyledons are fleshy, so these are the two cotyledons. This is a plumule. So, this part is a radicle, the root apex, and hypocotyle. So, these are the structures that we can observe in the dicotyledonous seed. Two cotyledons, plumule, hypocotyle, radicle, and the root apex. In this, the fleshy structures are present called as cotyledons, which are laden with a high amount of nutritive tissue. In monocotyledonous seed, single cotyledon is present. This cotyledon is generally peppery or thin, or sometimes it is a hard and shield-like structure. That hard shield-like cotyledon is called as scutellum. And in this, majority of the portion in the monocot seed is occupied by the endosperm. So, which is the part which will store the food materials in it. And in this monocot seed also, we can see the presence of epicotyle and hypocotyle. So, epicotyle, as we know that plumule part is present. The extreme tip of the plumule is covered with a sheet-like structure called as coleoptile, whereas hypocotyle extreme tip is called as radicle. So, this radicle is covered with a sheet-like structure called as coleorhiza. And in monocotyledon seed, the embryo part is pushed towards one lateral side, whereas majority portion of the monocot seed is occupied by the endosperm. And separating the endosperm from the embryo, there is an triploid layer is present called as alluron layer, protein rich layer called as alluron layer. So, if you observe the structure of this monocotyledonous seed, so like this we can see the structure of monocot seed. So, this part is called as a scutellum, and this is the plumule part, the coleoptile. So here root apex is present and coleorhiza, or root apex or root tip, coleorhiza, and the radical parts are present. So this is a little bit organization that can be seen in the monocotyledonous seed. So these are the changes which are taking place. Actually there is no, initially the changes will be same, but gradually when they start matured or when they convert into mature structures, little changes will be observed in the formation of the seeds of monocots and the dicots.